Okay, so we're going to discuss about the CSF or the cerebrospinal fluid. So speaking about the CSF, so CSF is the body fluid that try to flow here within our CNS. So CNS, this includes your central nervous system. It includes here the brain and the spinal cord. Okay, we have here the basic function of your CSF. So number one, provides here, try to remove metabolic waste in our CNS. The second one, try to provide nutrients to your brain and spinal cord. And lastly, we have here provides caution or protection with our brain spinal cord against the trauma. So our CNS is being lined here by three meninges. So we have here the outer, we have here the dura mater, which try to line directly your skull and your ventricles. And followed here by the next layer is your arachnoid. So arachnid, that's your spider-like na layer. And finally, we have here the pia mater, which try to line directly your brain and spinal cord. Okay, basically we have here the CSF is being produced here in the third and the fourth ventricles and try to flow here within your subarachnoid space. But basically, aside from your ventricles, your CSF is also, some of the volume of that is being produced here out of your plasma. Galing siya sa plasma natin or from your blood. So, kung ito yung bloodstream natin, tapos dito ang CSF natin, so pwede siya pumasok dito sa ating CNS. So, paano siya makapasok from your blood? If this one is your plasma, paano siya makapasok dito papunta sa CSF natin? That is through your selective filtration process. Pag sinabing selective filtration, so hindi lahat ng mga substances or your blood component here papasok sa CNS natin. If one is your CNS. So, kailangan mo muna i-filter ito. Okay? So, kailangan mo muna i-filter. Basically, this is being lined. Ang membrane nito para makapasok is being lined by the endothelial cell. Okay, the endothelial cell here is actually the one trying to provide your blood-brain barrier. Pag sinabing blood-brain barrier, hindi lahat ng mga plasma natin papasok sa ating CNS because meron siyang nafi-filter. So basically, paano siya makapasok? Through your hydrostatic pressure or active transport secretion. Pag sinabing active selective filtration, again, your component here of your CS, your plasma, kasi yung plasma natin may mga substances doon na like your medications, your WBC, dapat supposedly will not go to your CNS because they are harmful substances. So, para hindi sila makapasok, again, they are being regulated here by your blood-brain barrier, and that's being maintained by your endothelial cell, which try to provide here a very tight filtration membrane. Then, pag pumasok na dito sa CNS, again, hindi lahat, hindi pwede na pasok lang ng pasok ng plasma natin with your CNS. Dapat, some of that volume may lalabas din, pabalik sa ating blood circulation. And that we're going to be returned to your blood circulation, ang pumapasok dito, through your, lalabas siya through your arachnoid granulation or villi before it then eventually try to return to your blood circulation. That's to maintain here the normal na volume ng CSF. Kasi kung pasok ka lang ng pasok ng plasma ng volume, then eventually the dami ang fluid in your CNS is not normal. So therefore, may pumapasok, dapat may lalabas din. So basically, the CSF is being produced here at a rate of 20 ml per minute. In order for you then to maintain the normal values or the normal volume of the CSF within your CNS at a volume of 190 to 150 ml for the adult patient, and we have also 10 to 60 ml for the children. Okay, then we have here the collection of your CSF. So we collect, okay, the physician try to collect here the CSF through the process we call it was your lumbar puncture, where try to puncture the third, the fourth, the fifth, the vertebral column by a needle and try to collect the specimen. So basically, the volume of that being collected for that is highly dependent on the pressure introduced during the collection process. It could also be highly dependent on the existing medical conditions like meningitis, for example, and many other conditions here. So when you're collecting the specimen, then you need to prepare 3 tubes here. So that's 1, 2, and 3. And that's the number here is on the number of the collection process. Okay, so basically we have here the tube number 1. The specimen for that is being delivered in your chemistry and serology section. Okay, for your chem test and some serological tests. For the second tube, that's for your microbio, like your gram stain, your acid fast stain. And third one for your hematology, for your cell count. So our procedure for this one is when you receive the specimen, you need to process the specimen immediately. But if not, if there's a delay in the processing of the specimen, then for tube number 1, for the chem and serology, you could frozen the specimen until you are about to process the specimen. For the microbio, then you could have here your stored at one at room temperature. And for the hema, you could have put the specimen in the refrigerator prior to the processing of that. Okay, now we have here, we go to the appearance of the CSF. So the normal CSF is clear in color. Sometimes you could receive the specimen which is cloudy or turbid na specimen. And that might be because of the presence of high WBC count or high WBC in the specimen. That's signifying there is an infection. Like for example, we have here meningitis. And we have also here a milky specimen that will be associated here with your high concentration of your lipids. So we have the term xanthochromia, so specimen regarded here as a xanthochromic specimen, specimen with other color, like for example, it appears to be pink, orange, or yellow in color. So the pink here would have the presence of small concentration of your blood, your oxyhemoglobin. Then we have here heavy hemolysis would be related to your orange na color. And the yellow would be the presence of your bilirubin, the conversion of your oxyhemoglobin to become your unconjugated bilirubin. So other manifestation of xanthochromic specimen may be related here to the pigments. You could have your carotene. You could have your protein as well. So you have also here your malignant melanoma. And then another one, sometimes you could receive a specimen which is grossly bloody red na specimen. So if you receive the specimen like that, you need to differentiate if that one is caused here by traumatic tap or your intracranial hemorrhage. Kasi sinabing traumatic tap, that's being produced here once the specimen is being collected by your lumbar tap. So baka traumatic ang kanyang pagkuha ng blood, so become traumatic tap. Intracranial hemorrhage meant to say that's a bleeding already within the CNS. So even if you try to have your careful na collection, since may bleeding nga dun sa loob na, so still magkakaroon pa rin siya ng still bloody ang specimen natin, but not related to your mishandling procedure. So differentiate between your traumatic tap and your intracranial hemorrhage. Number 1, we have here the uneven color of your 3 tubes. So pag sinabing uneven color here, the first tube, siya ang pinaka-dark na red. So followed here by nagpi-faint. So ang ating red up to the tube number 3. So mean to say that one is your traumatic tap. But if the specimen, for example, here is even, so lahat ng tube mo, tube 1, 2, and 3, are puro lahat red, same intensity ng redness, then that's your intracranial hemorrhage. The second one, we have here the fibrinogen or the clot formation. So fibrinogen is being introduced when you perform your traumatic tap. Okay, sometimes you could receive a specimen naman na clotted pero hindi siya bloody red. It might related to your condition like your Freund syndrome, your meningitis, or blockage in the CSF. And sometimes could be clotted na specimen, hindi siya bloody. And then if you try to wrap the specimen, magpo-form siya ng parang spider-like or web-like pellicle formation. And that's related to your tubercular meningitis. Okay, next we have here the presence of the macrophages. The erythrophagocytosis is the macrophage we try to ingest the red blood cells. Basically, ang function ni macrophage natin, so remove your debris or even your RBC within your CNS. So, pag nakakita ka ng macrophage we try to ingest the red cells or macrophages, that's erythrophagocytosis, it signifies that the bleeding is already within the CNS even before you try to collect the blood. That signifies your intracranial hemorrhage. Of course, here, the D-dimer, the fibrinogenation products are the products of your fibrinogen breakdown. So therefore, it's related to your traumatic tap. Okay, the next one, we have here your cell count. So cell count being done with your CSF in the form of your total cell count. You could also have your WBC count. You could also have here your RBC count. But most likely, routinely, we are not performing the RBC count. So we just perform here the RBC count if the specimen is grossly bloody na specimen. Or you could also perform here indirectly obtain the RBC count. So perform mo lang ang total cell count. Okay, total cell count minus mo ang, ay sorry. Total cell count, tapos minus mo ang, ito lang gagawin mo. Total cell count, saka WBC count lang. Okay, then whatever the difference between the two that become your RBC count. So we have here the normal values, okay, for the total WBC count here. So that's 0 to 5. Okay, that's 0 to 5 cells here per microliter. Sorry, that's 0 to 5. Okay, those are 0 to 5 cells per microliter. But in the case of children, so mononuclear cells here, lymphocytes, especially the monocyte, will try to increase up to 30, still consider that was normal if your patient is among children. Okay, then we have here the procedure when you're performing your cell count. So again, if the specimen here is cleared, you don't need to dilute that one. And if that one is turbid ang specimen, you could dilute your specimen by the NSS, using your NSS. Okay, for your WBC count, so we have here 4 drops of your CSF. Then you just binuding here your WBC pipette. But first, you need to rinse the side of your WBC pipette with your 3% glacial acetic acid to lyse the red blood cells because you are only after of the WBC, or you don't need the obstruction here with the presence of your RBC para hindi mahirap mag-count and mag-identify. Then, sometimes you are adding methylene blue para magkaroon siya ng color that will help you differentiate your cells here. And then you count your cells in the 4 corner squares plus your central square in your Neubauer counting chamber. Okay, then we have here the formula for your WBC count. So we are still using here the usual formula in our hematology. So that's your cell count equals to your total number of the WBC counted in your 4 corner square plus your central square. Multiply that one with your dilution factor. Then we have the total squares counted. So you have 5 for that. Multiplied by the volume of each of the square. And another one, you need to have your necessary correction for the contamination because of the artificial introduction of your WBC. So sometimes here, pag traumatic ang puncture natin, we could introduce WBC during the collection process, which is not normally dapat part siya ng ating count here because again, artificially introduced lang naman siya because nag-collect ka ng specimen. You try to add that one during the collection process. It's not an actual na WBC present in the CSF. So for that, the formula for that, you need to have here the specimen, the CSF. Another one, you need also to collect blood from the patient. So what are going to do here? For the blood of the patient, perform WBC count. For the CSF, the patient, perform the RBC count. Another one, also perform here the RBC count for the blood of the patient. Okay, so this become here WBC added. So whatever the result, ima-minus mo doon sa nakuha mong result dito. Because again, should not be part of this total because total WBC count na na-count mo is because it's just only artificially introduced because nag-perform ka ng traumatic puncture. Or kung minsan, ayaw mo na mag-compute or gumawa nito, then you just need to derive. So, pwede mo itong gamitin if your RBC count ni patient sa blood niya saka WBC count ni patient sa blood niya are normal values or within the normal values. So, ito lang gagawin mo. In every 700 na RBC, with your RBC count, so minus 1 na WBC. So, ima-minus mo siya dito kung ano makukuha mo na result. Okay, then we have here the quality control. So, okay, we have here your diluent. Should be checked here twice a week for the possible contamination of your cells. Kasi kung minsan pag nag-perform tayo ng cell count, diba? So, pag nag-dilute, nag-aspirate tayo ng ating fluid, Kung minsan bumababa ang fluid natin sa ating diluent, it might contaminate your diluents already. So, kahit hindi mo pa siya nagamit, kung na-contaminate na siya, may mga cell na nandun, so therefore it become here added to your cell count. Therefore, you need to check that one. By examining, you get a sample of your diluent, examine that one under the microscope at 4x magnification. Another one, your centrifuge speed here should be checked once a month with the use of your tachometer to check here if that one is working properly with the speed. And we have also for your non-disposable na counting chamber, in your power counting chamber, then you need to rinse that one first with your bactericidal solution for 15 minutes, then rinse with the water, and end up drying with your isopropyl alcohol. Okay, the next one, we have here your differential count. So, pag sinabing differential count, we need to perform here your identification of your differentials. Okay, that will help us then to diagnose your conditions. Okay, so before you perform your differential count, you need to concentrate your sediments because sometimes here you just have a few na sediment, you need to recover that one. One, you could have performed here the sedimentation or filtration, pero hindi siya routinely ginagawa. Although that one procedure for your concentration here is okay siya because again, it would prevent here the distortion of your cell. Centrifugation, you could perform that one, but be careful with that. So you just have 5 to 10 minutes centrifugation, then supernatant is discard, then collect here the sediment at the bottom of your tube. You could also perform here the cytocentrifugation, which is best, but you need to add here your one drop of your 30% albumin. That's to prevent the distortion of the cell during the cytocentrifugation. Okay, so it's a conical tube. You add your specimen on that. Tapos is try to centrifuge, ma-recover na ang sediment with your 6 mm circle na slide. Okay, how you going to perform here your differential count? So after this, concentrating your sediment, okay, you try to transfer a drop of your specimen sediment here in your slide and then make a smear, air dry, then you stain, and then try to identify the different cells. Okay, and then classify that and then report that one in terms of the percentage. Like, ilan ang neutrophil, ilan ang lymphocyte. So, 100 dapat i-count natin. And then, try to present that one in terms of the percentage. Okay, on the other hand, you need also to perform here your quality control with your slide for possible bacterial contamination. So, for the slide here, 0.2 ml of your saline. Okay, then we have also here your 2 drops of your albumin. Okay, for possible slide contamination of your bacteria, then perform that 0.2 ml of your saline, 2 drops of your albumin. Okay, then try to examine here for the slide for possible bacterial contamination.